In this part, we will discuss motilities along gastrointestinal system. Once you have introduced the bolus of food into mouth, you are starting the first movement, which is chewing or mastication. That movement is voluntary, but it's having also reflex behaviors. The purpose of this movement is mixing food with saliva and grinding that food to smaller particles. So after mastication or chewing, there is the second also movement that is starting, which is deglutition or swallowing. That's the process of deglutition. So the bolus of food, after mixing it with saliva, is pushed from the mouth toward the pharynx with the help of tongue. Now the pharynx is having many openings. One is toward nasal cavity, one is toward trachea, one is toward esophagus, in addition to the one which is toward the mouth. Now, that opening toward the mouth is closed with the help of the tongue. The opening toward the nasal cavity, the back opening of the nasal cavity will be closed with the help of the soft palate. Trachea will be closed with the help of epiglottis. Now, after these reflex mechanisms, which are taking place as reflex behaviors, there will be a contraction of the pharynx. In addition, the upper opening of the esophagus is guarded by a sphincter, which is called upper esophageal sphincter. So this sphincter will relax. So by the contraction of the pharynx and by closing all these openings, we are forcing the bolus of food to move into the esophagus. So if you are analyzing the process of swallowing, that process is initiated as a voluntary act and then continuing with involuntary reflexes. As a voluntary act, you are pressing or pushing the bolus of food from mouth into the pharynx. Then you have at the pharynx a lot of reflexes which are resulting in closing of the back opening of the nasal cavity, closing of trachea with the help of epiglottis, relaxing the upper esophageal sphincter, and contraction of the pharynx. The contraction of the pharynx also can have a place as a voluntary action. So, we are analyzing the swallowing. It is having a voluntary stage and is having also involuntary stages. As in voluntary stages, there is a pharyngeal phase as an involuntary state. Also, there is an esophageal phase as a voluntary stage. Now you are continuing the process of swallowing along the esophagus. We have movements also along the esophagus which are conducting the bolus of food toward the stomach. Now this is a process which is the esophageal phase. So what happens at the level of the esophagus? They are motor movements which are called esophageal peristaltic contractions. These esophageal peristaltic contractions appear as a contraction up to the bolus of food, relaxation down to the bolus of food. By that movement, the bolus of food is forced to move forward toward the stomach. So this is showing you the esophageal peristaltic contractions. You have contraction up and relaxation down. Now the bolus of food reaching the lower part of the esophagus. That lower part of the esophagus is also guarded by another sphincter which is called lower esophageal sphincter. What happens to that sphincter? This sphincter will relax by reflexes, which are intrinsic reflexes through the enteric nervous system. Now, for the purpose of understanding of the swallowing along the esophagus, we have two types of peristaltic contractions at the level of the esophagus. During esophageal phase of swallowing, there are two types of peristaltic contractions and called primary esophageal peristaltic contractions and secondary esophageal peristaltic contractions. Both types of peristaltic contractions are having the same pattern. What's the difference between the primary and the secondary? If you remember, the process of swallowing or the peristaltic contractions along the esophagus are continuing after the contraction of the pharynx. So, if that wave of contraction is continuing with a wave of peristaltic contraction along the esophagus, we are calling this type of peristaltic contractions as primary esophageal peristaltic contractions. But sometimes happens that the primary esophageal peristaltic contractions are failing to conduct the bolus of food toward the stomach. So, at any time that food is remaining at the level of the esophagus, it can reinitiate the same pattern of contractions, but we are calling that type of peristaltic contraction as secondary esophageal peristaltic contraction. So the difference between primary and secondary peristaltic contraction is the initiation, or at what organ we have initiated these contractions. The primary have been or have following the contraction of the pharynx as a wave of contraction behind the bolus of food, while the secondary, they can be initiated at the esophagus itself if the primary has failed to conduct the bolus of food toward the stomach. Now, what happens at the level of the stomach? Once we have that food is reaching the stomach, what happens at the level of the stomach? First, there will be a relaxation. The stomach, when it is empty, is having very low capacities, only 50 to 100 milliliters. But you can fill your stomach with food, fluids, and so on. And by filling that stomach with food, there will be a dilation of the stomach. That dilation is called receptive relaxation of the stomach. The receptive relaxation is a decrease in the tonic contraction of gastric smooth muscle cells. Over that receptive relaxation, we have another motor activity which is taking place there, and called gastric peristaltic contractions. What that gastric peristaltic contraction is? The gastric peristaltic contraction appears as a wave of contraction which starts from the upper part of the stomach, traveling along the whole body of the stomach until reaching the pyloric sphincter. That wave of contraction is like a constrictive ring at the wall of the stomach. Now, once it is reaching the pyloric sphincter, that sphincter becoming more constricted, so more closing of that pyloric sphincter and not opening. So what happens by more closing of that sphincter? The content between the constrictive ring and the pyloric sphincter, most of that content is tossed back toward the stomach. We can have small amounts of the content which can be emptied from the stomach into the duodenum, but most of the content is tossed back. So these are the effects over the content of the stomach. The amount of the content which is tossed back is having an effect which is called retropulsion, while the amount which has been emptied from the stomach into the duodenum through that smaller opening must be of fluid consistency, and it is called gastric emptying. So what happens at the level of the stomach? During gastric phase, the gastric content is moving between the lower part of the stomach and tossed back toward the stomach, so it's having that effect which is called retropulsion. The purpose of that movement is to mix food with the secretions from the stomach, and by mixing that food with the secretion, we are not having the content of the stomach as food. It is now called chyme. And we have a small amount that can be emptied, but that amount which is emptied, it must be of a fluid consistency. Some they are calling that activity of the pyloric region, which is the lower part of the stomach, as pyloric pump activity. So by the pyloric pump activity, we have retropulsion, and we have small amount of chyme can be emptied into the duodenum. The last type of movements at the level of the stomach, they are called hunger contractions. These contractions are taking place between meals when the stomach is empty and when you are hungry. How they are initiated, nobody knows, but there is a relation between the decreased glucose level in blood and the initiation of the hunger contractions. Gastric movements are controlled by neural control and hormonal control. The neural control is the main controller for gastric motility. We have the enteric nervous system. We have also the autonomic nervous system, which can increase gastric movements by the distension of the stomach. So that local changes or the distension of the stomach activates the enteric neurons, and the neurons of the autonomic nervous system of the vagus nerve to increase the motor activities of the stomach. Once we have emptied the content of the stomach into the duodenum, that emptying causing distension of the duodenum. That distension activates enterogastric reflexes, which are inhibitory reflexes. These reflexes are through the parasympathetic system. So now during intestinal phase, so once we have the chyme reaching the small intestine, the gastric activity is inhibited by these reflexes. In addition to the neural control, we have hormonal control. Gastrin, which is released from gastric mucosa, as you will see in secretion, it can increase gastric activity. But we have also some hormones which are released by the mucosa of the small intestine, like cholecystokinin, secretin, GIP, gastric inhibitory peptide. All these can act over smooth muscle cells of the stomach to decrease their motor activity. So these hormones, the cholecystokinin, secretin, and the gastric inhibitory peptide, are inhibiting gastric motor activities. At the level of the small intestine, many motor activities are taking place. We have segmentation contractions, peristaltic contractions, migrating motor complex contractions, peristaltic rush. The rhythmic contraction of the circular layer is giving the segmented appearance for the small intestine. And because of that appearance, we are calling these types of motor activities as segmentation contractions. These segmentation contractions are rhythmic. So if you are viewing the point where you have contractions of the circular layer, after a while that point will be in relaxation, then again will be in contraction, and so on. The segmentation contractions are following or having the same rhythm as the slow waves. They are about 12 contractions per minute in the upper part of the small intestine, and there are about 8 contractions per minute in the lower part of the small intestine. Also the slow waves in the upper part of the small intestine, they are about 12 slow waves per minute, and in the lower part they are around 8 slow waves per minute. What is the effect of these segmentation contractions over chyme? Mixing, mainly mixing. So it's mixing chyme with secretions and enzymes that are released from the pancreas, as example, secretion from the small intestine and secretion from the pancreas, and so on. So that's the effect of segmentation contraction over chyme. They are having mixing effect over chyme. The second type of contraction, they are called peristaltic contraction. As you see, it is a contraction up, relaxation down. Between the points where you have contraction and relaxation, there is also all the time rhythmic activity of the longitudinal layer. So there is shortening and elongation. The main effect of the peristaltic contraction over the chyme, which is the content of the small intestine, is to propel that content in analward direction. So it is having propulsive effect on chyme. So we have seen the segmentation contractions are rhythmic contractions. So the main control for these rhythmic contractions is the electrical control. Also, in peristaltic contraction, we have the shortening and elongation of any segment along the small intestine, which is also rhythmic contraction. So that part is also controlled by the electrical activity. In addition to the control by the electrical activity, at any time you have contraction up, relaxation down, that control is achieved by the enteric nervous system. Usually, the enteric neurons, the excitatory neurons are projecting up and the inhibitory neurons are projecting down. So at any site you have excitation, you are getting up contraction and down relaxation. Also, we cannot exclude effects for the autonomic nervous system over intestinal movements. The parasympathetic can increase intestinal movements while the sympathetic can decrease these movements. Also, we can have some hormonal control over intestinal activities like gastrin, cholecystokinin, serotonin can enhance intestinal movement while secretin and glucagon can inhibit intestinal movement. But the main control after the electrical control, which is controlling the rhythmic contractions, is the enteric nervous system and the autonomic nervous system. To a lesser extent, we have the hormones can affect movements, but they are not main controllers for the movements. So we can summarize motilities of the small intestine. We have segmentation contractions, and the main effect of that segmentation contraction is mixing effect over chyme. But we cannot exclude some propulsive effect for the segmentation contraction. Peristaltic contractions, the main effect is propulsive, but we cannot exclude some mixing effects for the peristaltic contraction. Finally, by the effect by propelling the content or chyme along the small intestine, that content is moving at a velocity of 1 centimeter per minute. So every minute we have chyme moving along the small intestine by 1 centimeter. In addition, we have migrating motor complex contractions, which can take place between meals when the small intestine is empty. So they are starting as contractile ring, starting from the stomach, traveling along the whole small intestine. The purpose is to clean out, to sweep out everything from the small intestine after the process of emptying the small intestine into the colon. So that migrating motor complex, they are cleaning the content of the small intestine. In addition, in some pathological conditions, if there are harmful pathogens invading the small intestine, they are activating the powerful peristaltic contractions, which are called peristaltic rushes. So, as symptoms, they are appearing as diarrhea, watery diarrhea, and so on in that person. At the level of the colon, there are two types of motor activities. There are rostral contractions, which are present all the day over the colon, and there are mass contractions, which can happen only once, twice, or 3 times a day, a series of contractions for about 15 to 20 minutes, and after 20 minutes they are dying. In the colon, we have one variation with regard to the longitudinal layer. Smooth muscle cells, which are forming the longitudinal layer, are grouped together to form 3 strips around the colon, which are called tinea coli. By the tonic activity of tinea coli and by the rhythmic activity of the circular layer, we are getting this appearance for the colon, which is like the segmentation at the level of the small intestine. These bulges, which are resulted by the activity of the circular layer and by the higher tone of the tinea coli, the strips around the colon, these are called rostra, and because of that we are calling this type of motor activity as rostral contractions. These rostral contractions are having propulsive effect over the content of the colon. They are responsible for moving the content from cecum and ascending colon toward the transverse colon. And to move that content, there is a need for about 9 hours. That time is useful for getting more reabsorption of fluids from the content of the colon. The content in the transverse colon is becoming more solid content. To move that more solid content after removal of water and electrolytes toward lower structures of the colon, there is a need for a more powerful movement. That more powerful movement is called mass contraction, which can move the content from the transverse colon toward the descending and sigmoid. These mass contractions are having propulsive effect, but they are not found all the day over the colon. They can take place only once, 2 times, or 3 times a day, mainly after getting breakfast. So, after getting breakfast, there is a distension in the stomach. That distension is initiating reflexes, which are called gastrocolic reflexes, to initiate the development of mass contractions. Now, by the activity of mass contractions, the content from transverse colon and descending colon is propelled toward lower structures like sigmoid. By having more fecal materials in sigmoid, there is a distension in that organ. This distension will activate what we are calling intrinsic defecation reflexes, which means there will be higher motor activities by that distension and by the activation of enteric nervous system at that level. By having these intrinsic defecation reflexes, there is a movement of fecal materials toward lower structures, toward the rectum. At the rectum, there is activation of extrinsic defecation reflexes. So there will be an activation of the autonomic nervous system, more precisely sacral fibers or parasympathetic fibers from sacral origin are activating more motor activities at the rectum. That more motor activities at the rectum may end finally with the defecation act. But the defecation act can take place only by relaxation of sphincters. There are two sphincters which are guarding the anus. These sphincters are internal anal sphincter and external anal sphincter. The internal anal sphincter will relax by these reflexes, extrinsic and intrinsic reflexes, while the relaxation of external anal sphincter is taking place as a voluntary relaxation. It is under the control of the somatic nervous system. The fibers of external anal sphincter are striated muscle fibers, so they are under the voluntary control. By the relaxation of the two sphincters and by the higher activity of the rectum, there will be a defecation act which can take place. Sometimes the fecal materials are more solid. There is a need for higher pressure inside abdomen. That can be achieved by holding respiration and by contraction of the abdominal muscle, which results finally in the defecation act. So finally, to get defecation, there will be activation of intrinsic reflexes and extrinsic reflexes. The intrinsic reflexes are through the enteric nervous system. Extrinsic reflexes are through parasympathetic fibers from sacral origin, which finally results in increasing motor activities at the level of the rectum and relaxation of internal anal sphincter. But also the defecation act can take place only by the relaxation of external anal sphincter, which is under the voluntary control. When that person is at the right time, right place, he can go to the bathroom and having defecation act.